Quality control is an essential operation in the pharmaceutical industry, and in this video, as a part of our pharmaceutical technology project, we will discuss quality control of the liquid dosage forms. Let's start with the definition of quality control. Quality control is a procedure or a set of procedures that are designed to ensure the output of uniform batches of the drug conforms to the established specifications of these formulations. Different dosage forms require different quality control tests, but there are some tests that will be performed for each liquid dosage forms regardless of their type.
In visual inspection, ingredient and the final product are carefully examined for purity and for appearance. Physical appearance of a product for patient adherence and compliance is critical, so should be good looking and elegance in appearance.
Color, odor, and taste are very important parameters for the orally administered formulations. Porability measurement that used to ensure the porability of the final products and it doesn't cause any problem during the filling or the handling by the patient.
Viscosity measurement. Viscosity is property of liquid that's directly related to resistance to flow. Viscosity measurement is very important quality control test in case of syrup and elixirs. Viscosity directly relates with stability of solution. The higher viscosity, the greater stability.
There are different methods that can be used to measure viscosity of a liquid. When we use a device to measure viscosity, we call it a viscometer, and we have two types of the viscometers: cone and plate viscometer, cup and pump viscometer, and we choose one of them depending on the formulation that we use and what the monograph tells us.
Determination of viscosity of liquid using Ostwald viscometer. It's U-shaped glass apparatus with wide arm and narrow one with capillary tube in narrow part. There are two marks above and below the bulb in narrow part. In this method, the viscosity of liquid, viscosity 1, can be determined in relation to another liquid known viscosity 2. The viscosity of the liquid under test, viscosity 1, can be found by measuring the time 1 required for this liquid to pass between the two marks. Is it flows by gravity through the vertical capillary tube. This time is compared with the time 2 required for liquid of known viscosity 2, usually water, to pass between the two marks. Procedure: pour water into the wide arm. Suck water from the narrow arm till rise to the upper mark. Close the top of narrow arm with your finger and inflate the viscometer to empty the wide one. Record the time taken by water to move from the upper to the lower mark. Repeat the last step twice again and find time 2 and time 3. Then calculate mean time. Repeat the previous steps for liquid to be measured and fill the table of result. pH measurement. The measurement and maintenance pH is also very important step in quality control testing. General, there are two different types of method used in measurement of pH: pH paper and pH meter. pH paper method. The simplest and cheapest is to dip a piece of pH paper into the sample. The paper is impregnated with chemicals that change color and the color may be compared to chart supplied with the paper to give pH of the sample. pH meter. If greatest accuracy is required, pH meter should be used. A typical pH consists of a special measuring glass electrode connected to an electronic meter that measures and displays the pH reading. Importance of pH: drug solubility and absorption. pH can affect solubility of drugs. Weakly acidic drugs are more soluble in alkaline solution. Converted into salt form, more soluble. If pH is lowered, acidic drug will precipitate. Weakly basic drugs are more soluble in acidic solution. If pH is raised, basic drug will precipitate.
In addition to all the previous quality control tests that we discussed, there are some special tests for each dosage forms, and we will start with the suspension quality control tests. The sedimentation volume. Sedimentation is the process of allowing particles in suspension in water to settle out of the suspension under the effect of gravity, and it's expressed by the ratio of ultimate volume of the sediment and the original volume before the sedimentation. And it can be measured by preparing a series of different suspensions of different suspending agents or different concentrations of the same suspending agents in different time intervals. Degree of flocculation is the ratio of the sedimentation volume of the flocculated suspension to the sedimentation volume of the deflocculated suspension, and it's more fundamental parameter than sedimentation volume since it relates to the volume of the flocculated sediment to that in the deflocculated system. If the flocculated sedimentation volume is larger than 1, we can say that the system is in flocculated equilibrium. Moving to the emulsion quality control tests, measuring the thermal stress can help identify the likelihood of degradation of products by subjecting the product to high and low temperature, helping to evaluate the emulsion stability and establish degradation pathways. Phase separation is one of the most important parameters to be measured for the emulsions. It can be easily determined by using a graduated cylinder and measuring the volume of the separated phase after a time interval. This picture here shows different types of phase separation that can occur in the emulsions. Another important parameter that used for the evaluation of emulsions. Particle size and count can be performed either by the optical microscopy or by measuring the sedimentation. If the emulsion particle size range from 0.2 to 100 micrometer, we use the optical microscopy. Another method is to measure the sedimentation of the emulsion. First, prepare the emulsion and place it in the sedimentation vessel. Then take 10 milliliter sample at specified intervals of time and depth. Dry the sample. Then start comparing the samples of different time intervals in terms of the particle size and count. Electrophoretic properties. The determination of the electrophoretic properties, such as the zeta potential, is very useful to assess the flocculation or rate of the emulsion, especially if the particle size is large. This table shows the effect of the zeta potential value in the stability of the emulsions. As an example, if the zeta potential is more than 61, it means that we have an excellent stable emulsion.
Quality control test that we will cover for parenteral dosage form, sterility test, biogen test, and particulate matter test. What are parenterals? Parenterals are a sterile dosage form intended for administration other than internal route and exert their action by directly entering into systemic circulation. Test for sterility. Sterility is defined as freedom from the presence of viable microorganism. It has two methods. Method A, membrane filtration. Method B, direct inoculation. Medium to be used in the sterility test: fluid thioglycolate medium, soybean cyazyne digest medium. Membrane filtration method. A membrane has a nominal pore size not greater than 0.45 micro and diameter of approximately 50 millimeter. This method basically involves filtration of sample throughout membrane filters. The filtration is assisted under steric aseptic condition. After filtration complete, the membrane is cut into two halves, and one half is placed in suitable volume of 100 ml, usually fluid thioglycolate medium and soybean cyazyne digest medium. Incubate the medium for not less than 14 days. Direct inoculation method. The direct inoculation method is the more traditional sterile test method. Basically, the direct inoculation method involves 3 steps. 1. Aseptically opening each sample container from a recently sterile batch of product. 2. Using a sterile syringe and needle to withdraw the required volume of sample for both medium from the container. 3. Injection 1/2 of required volume sample into a test tube containing the required volume of a fluid thioglycolate medium, and the other half volume of sample into a second test tube containing the required volume of a soybean cyazyne digest medium. Minimum quantity to be used for each medium. If a liquid less than 1 ml, the whole content of each container. If a liquid from 1 to 40 ml, have the content of each container but not less than 1 ml. If a liquid greater than 40 ml and not greater than 100 ml, 20 ml. Greater than 100 ml, 10% of the content of the container but not less than 20 ml. Interpretation of result. If the materials being tested renders the medium turbid so that the presence or absence of a microbial growth cannot be easily determined by visual inspection, 14 days after the beginning of an incubation, transfer portion less than 1 ml of medium to fresh vessels of the same medium, and the incubate original and transfer vessels for not less than 4 days. If no evidence of microbial growth is found, complies with test for sterility. If evidence of microbial growth is found, does not comply with test for sterility. Test for albirogen. The test involves measurement of the rise in body temperature of rabbits following the IV injection of a sterile solution into air vein of rabbits, does not exceeding 10 ml per kilogram injected intravenously within a period of not more than 10 minutes. Test animals. Use healthy adult rabbits of either sex, preferably of the same variety. Recording of temperature by clinical thermometer. Preliminary test. Shame test. If animals are used for the first time in a biologen test or haven't been used during the 2 previous weeks, condition them 1 to 3 days before testing the substance by injection IV 10 ml per kilogram biologen free saline solution, warmed to about 38.5 degrees Celsius. Record the temperature of the animals beginning at least 90 minutes before injection and continuing for 3 hours after injection. Main test. Procedure. Carry out the test using a group of 3 rabbits. Preparation of the sample by dissolving the substance in or diluted with biologen-free saline solution. Warm the liquid to approximately 38.5 degrees Celsius before injection. Inject the solution under examination slowly into the marginal veins of the ear of each rabbit over a period not exceeding 4 minutes. Record the temperature of each animal at half-hour intervals for 3 hours after injection.
the difference between the initial temperature and the maximum temperature, which is the highest temperature record for a rabbit, is taken to be its response. Interpretation of result. If a number of rabbits, 3 rabbits, and individual temperature rise 0.6 and temperature rise in group 1.4, test passes. If above not passes, add 5 and the total become 8 rabbits. And individual temperature rise 0.6 and the temperature rise in group 3.7, test also passes. Particulate matter test. Particulate matter refers to the extraneous, mobile, and dissolved particle other than gas bubble, unintentionally present in the solution. 2 methods are used. 1. Light obstruction particle count test. 2. Microscopic particle count test. Light obstruction particulate count test. Use a suitable apparatus based on the principle of light blockage, which allows an automatic determination of the size of particles and the number of particles according to size. How is particulate matter testing using light obstruction performed? Light obstruction particulate matter testing uses a liquid particle counter machine that, based on the principle of light scattering, allows for an automated determination of the size of particles in a sample and the number of particles according to size. The liquid particle counter is calibrated using dispersion of spherical particles in particle-free water. These spherical particles are known size between 10 micrometer and 25 micrometer. Before use, all glassware and testing equipment are rinsed with particle-free water. Samples of particle-free water are tested. Following equipment set up to assure that no more than 25 particles of 10 micrometer or greater are identified in particle-free control and replicate. If more particles are identified, the test equipment will be cancelled again and replicate before sample testing. Limits. If a sample large volume parental greater than or equal 100 milli and the particle size 10 to 25 micrometer, maximum number of particle 3 to 25 in 1 milli. If a sample small volume parental 100 milli and less than 100 milli and the particle size 10 to 25 micrometer, maximum number 600 to 6000 in the container. Microscopic particle count test. Procedures. With the inside of the filter holder fitted with the membrane filter with several millimeters of particle-free water, transfer the total volume of a solution bowl or of a single unit to the filtration funnel and apply vacuum. Place the filter in a petri dish and allow the filter to air dry. After the filter has been dry, place the petri dish on the stage of microscope. Scan the entire membrane filter under the reflected light from the illuminating device and count the number of particles. Limits. If a sample large volume parental greater than or equal 100 milli and the particle size 10 to 25 micrometer, the maximum number of particle allowed is 2 to 12 in 1 milli. If a sample small volume parental 100 milli and less than 100 milli and the particle size 10 to 25 micrometer, the maximum number of particle allowed is 300 to 3000 in the container.
Elixirs. Elixirs are sweetened hydroalcoholic liquids for oral use. They usually contain flavoring and coloring agent to improve patient acceptance. Elixirs are better able than aqueous syrup to maintain both water-soluble and alcohol-soluble components in solution. Elixir quality control. Determination of alcohol content. Determination of alcohol content. Elixirs provide the capacity of the distilling flask that's sufficient, commonly 2 to 4 times the volume of liquid to be heated, and the rate of distillation is such that clear distillates are produced. Cloudy distillates may be clarified by agitation with talc or with calcium carbonate, and filtration is done, after which the temperature of the filtrate is adjusted and the alcohol content determined from the specific gravity. During all manipulations, take precautions to minimize the loss of alcohol by evaporation, for liquid is presumed to contain less than 30% of alcohol. Quality control test for syrup. Transmittance of light, visual inspection, pH measurement, sucrose concentration, physical stability in syrup, viscosity. Syrup, concentrated solution of sugar, such sucrose in water or other aqueous liquid, sometimes with medicinal agent added, usually used as flavored vehicle for drink. It's commonly expanded to include any dosage form, oral suspension, in sweet and fast fecal. Transmittance of light. Light transmittance meters is a newer tool that's used to check syrup color. Syrup sample is checked for color by passing light through the sample. The percent of light transmission is compared to light transmission rates for different grades. When using one, you need to be sure there are no fingerprint in test bottle and no bubbles in sample.